Hello students and welcome back to another class. Now today's topic is on northern and western blot. So in previous lectures we have already covered about southern blot, and that was specific for different types of DNA sorting, different types of not types we can say, but different sizes of DNA, and you can say all applications we also saw in that. Now today's lecture is specifically on northern and the western blot. So northern blot is specific for RNA detection and western blot is specific for protein detection. Now most of the things that we have learned is same, but a little differences. Specifically the western blot is quite different because we'll see that apparatus is also different that is used for blotting, as is seen in the DNA and RNA that the blotting is usually done with the help of diffusion of the buffer, but in western blot we use a specific apparatus for that, that is your gel electrophoresis apparatus only, where we have to apply a external current for the blot to transfer. So that we'll see. So let's start our lecture with northern blotting. So northern blotting as I told you that this blotting is specific for RNA detection. So here RNA detection takes place. Okay, apart from that, in the previous section, that is in DNA southern blotting, we saw that it is specifically their restriction enzymes are used. Here formaldehyde is used. Formaldehyde is used. Now restriction enzymes can be used if the RNA length of the RNA is very long that you are quantifying or you want to analyze. Otherwise formaldehyde gel is used, also known as denaturing gel, because different types of RNAs can be there, so first they have to be denatured properly. Okay, so yes, northern blotting is specific for RNA detection, then formaldehyde is used, and it is also, the northern blotting is also used for transcriptional analysis. We'll see that in application also. Transcriptional analysis. So now again, if you have a cell, if you have this cell and inside the cell you have this RNA, so first you have to homogenize the cell properly. Homogenize it. Then after homogenizing you have to extract the RNA. Now this can be done through various processes like centrifugation, density gradient centrifugation, and other types of process. Once the RNA is in your hands, now you have to run it on gel. Okay, so you have to perform gel electrophoresis. And once this is done, we have to perform blotting. So as I told you that the procedure is more or less similar, only that specific like formaldehyde is used here, that is different. So let's see. So we have already RNA in our hands, and now we have to just do gel electrophoresis. So after gel electrophoresis, you know that RNA also consists of negatively charged backbone as similar to DNA. So in the gel electrophoresis apparatus, it will go from negative to positive side. Here are your wells. Okay, so one will be your ladder that will give you all the types of RNA molecules of different molecular sizes, so that you can just compare it with your RNA molecule. And then you have, for example, you are doing transcriptional analysis in which in the cell all different types of RNAs are there, and you want to detect specific RNA or mRNA, that if it is present or not, and the quantity of it. So for example, you also get this type of bands a lot of bands you will get with different band sizes, which also shows its concentration. Okay, and the more away from the negative side, the lesser is the size of the RNA, or the lesser is the molecular weight, because it travels easily. So for example, this type of gel you have got. Now after this gel in your hands, now what you will do? Now we will do blotting. So in the blotting, let's draw the apparatus quickly. So this will be your plate, glass plate, in which you will fill your running buffer. Yes, buffer. And then here you will first keep some type of, okay, this is not visible, some type of base, sponge maybe, and on this sponge you will keep certain tissue papers so that it will absorb the buffer. And on this tissue paper you're gonna keep the gel. Here is your gel, and then you keep your nitrocellulose membrane. So let me select